Okay, good morning, everyone. So before we start with today's lecture, a quick reminder, you will have a homework assignment this week due on Friday that discusses tumor suppressor genes, the things that we've been talking about this past week and today's lecture as well. And you also have a quiz due on Wednesday at normal quiz time, meaning it's due right at our class time, 10:30 on Wednesday. So keep an eye out for those. I will post those later today, as well as sending you a reminder. Okay. All right, so let's continue our discussion about tumor suppressor genes and their importance in understanding how cells lose control over things like oncogenic pathways. So to remind ourselves of where we're at, because I think the last time we had a lecture was last Wednesday, we've been discussing tumor suppressor genes, or TSGs, and these are genes that code for proteins that act against certain signaling pathways that are involved in things like proliferation, survival, or motility, all of those things that we would associate with oncogenic, transformative processes unless they're kept under control. Right? And so the TSGs keep these types of pathways tightly under control. We've looked at some examples, such as PTEN. PTEN was the first one that we looked at. PTEN discourages mitosis from happening by interfering with the PI3 kinase signaling pathway that activates AKT. We also looked last time at retinoblastoma, right? Another example of a important tumor suppressor gene that prevents the cell from moving through the cell cycle and keeps the cell in G1, where the cell normally wants to do its normal differentiated cell functions, right? And so we learned that RB does that by resulting in the phosphorylation or the sequestering of different types of transcription factors, right? And once RB is phosphorylated, RB no longer can hang on to these transcription factors, and these transcription factors then seek out their various gene targets, the promoters, enhancers of certain genes that are associated with the cell moving into S phase. Right? So these tumor suppressor genes, whether you're talking about PTEN or retinoblastoma, they discourage mitosis from happening or the cell even wanting to go to mitosis, right? Unless the cell receives stronger proliferative messages from the outside, right, such as growth factors that signal through RTKs. And if the balance shifts and those pro-proliferation signals win out over the function of these tumor suppressor genes, that's what makes the cell then go through the cell cycle. Right? We also learned that inherited mutations in certain tumor suppressor genes, like RB, for example, that might happen through a point mutation or a chromosomal deletion, can explain certain familial cancers. In other words, the reason why a child is at greatly elevated risk of developing a particular cancer compared to the normal population, right, may be due to the fact that he or she inherited one bad copy of something like RB or PTEN. Right? And that's why those cancers can be followed through family generations. We also describe this important concept of loss of heterozygosity. So I always, thought that this term LOH was a little counterintuitive, right? Because loss of heterozygosity, for some reason, I couldn't wrap my head around it when I was first a graduate student learning about this. But really, it just means in a situation where you already are down to just one good copy of a gene and the other copy is somehow compromised, for example, through inheriting a bad RB allele, right? That individual is heterozygous at that locus, right? They would be RB plus minus, right? So loss of heterozygosity has to do with the plus also now getting a hit and becoming a minus. So now that you've become homozygous for the minus, right? That's loss of heterozygosity. So a cell that has one good copy of a compromise, or one copy that's good and one that's compromised, and then proceeding to lose the other good copy. So I want you to consider this as sort of a thought question. Right? Inherited mutant tumor suppressor gene alleles can be thought of as being recessive at the individual cellular level, and yet they are absolutely dominant at the whole organism level. What do we mean by this? Right? I think just the way this sounds has this kind of like, riddle kind of sound to it. How can something like a mutation be recessive or considered recessive at the cellular level and yet dominant at the whole organismal level that contains that cell? So you think about that. And as a hint, think about what we talked about, a child born with familial retinoblastoma and what takes place inside of his or her body. Right. Let's talk about different ways that tumor suppressor genes can have their expression negatively impacted. Right. So DNA methylation is a very important mechanism by which the cells of your body control whether certain genes are shut down or allowed to be transcribed. So DNA methylation is not something that's done in necessarily a very dynamic moment-to-moment fashion. Right? The methylation of certain groups of genes is usually associated with those genes no longer being needed for the particular cell that you're talking about. Right? So whole sets of genes that are no longer relevant to being something like a nerve cell or a muscle cell or a gland cell, right? Those cells might just keep them kind of shut down in a more permanent fashion. Because remember, all of those cells still have the genes in them. All of your nucleated cells still have your entire master blueprint copy of the genome, all 25,000 genes or whatever, right? It's just some of them never will need to use certain genes ever again once they've become a fully differentiated cell, right? And so they can kind of more or less permanently shut certain genes down. And that's what DNA methylation is about. So in our DNA sequences, we have every so often a C right next to a G on the same strand, right? So they're connected by the phosphodiester bond. So that's why you can say, you can put a little P in here to remind yourself that this is what we're talking about, a C next to a G on the same strand, not a C that's base pairing with a G across the strand, right? That's why the little P is a good reminder. So these CpG sites, okay, can be methylated. In other words, if you start off with a cytosine, you can have a methyl group added to them, right? A CH3, so you end up with 5-methylcytosine. Oftentimes, these 5-methylcytosines, because they resemble thymine, right, they can be deaminated. In other words, if this amine group is removed and you still leave the methyl group here, I mean, even though it would have been better if they still showed the CH3 here, but I think you know what that's what that means, right? By deaminating the 5-methylcytosine, you actually end up with a thymine. So as part of DNA repair, oftentimes, right, a methylated cytosine, as your cell tries to figure out, wait, what is this? What is this supposed to be? Is it supposed to be a cytosine? Well, it actually looks more like a thymine, right? So you have enzymes that deaminate this, and it actually turns it into a thymine. That means over evolutionary history, right, sites in our genome that were CpGs, many of them have turned into TPGs now. When we look at this on the right side, so we have two different sets of genetic sequence here. On the right side, this is sort of your typical DNA in a random location in your genome. Like if you closed your eyes and put your finger down on a chromosome and then looked at what the sequence is, chances are you're not going to be, at a promoter, but you're probably put your finger down in some intergene region, right, in between where genes are, or maybe you've ended up kind of being lucky and stuck your finger right in the middle of a coding region of a gene. This is what it would look like. You would see that proportionately, there are far fewer CGs in the human genome than you would expect by probability alone, right? Probability is what's the chances of having a C next to a G if your genetic sequence is random? Well, it's a 1 in 4 chance times a 1 in 4 chance, a 1 in 16 chance, right? So that would suggest that every 16 random dinucleotides that you put your finger on, right, 1 out of 16 of them would be a CPG. But in reality, we have far fewer than that. Unless you did put your finger down in the promoter region of a gene, right? Because you can see here on the left, this is a typical promoter region. Here's the ATG, so that when the RNA transcript, right, is made, ultimately this would be the translation start, where the ribosome would then start reading and translating the signal, right? So this region upstream of this ATG would be somewhere in here considered like the promoter region, right, where RNA Pol II is going to bind here and start transcribing. And you can see that it is rich in CPGs. In fact, you have many CPGs that are just strung together, conserved by evolution, right? So in other words, evolutionary history has favored that in these cases, at the promoters of genes, those CPGs are not lost. They are not turned into TPGs, right, like you find elsewhere in the genome. These are sites of DNA methylation, right, where those cytosines are allowed to be methylated, resulting in what? The DNA's shape being ever so slightly altered so that when they're wrapped around nucleosomes, right, the histone proteins that make the nucleosomes, they hold on much tighter to the DNA, right? And if the DNA is more tightly held around the nucleosomes and you have a string of nucleosomes that are all tightly holding onto the DNA, the chromatin becomes condensed. And if it's condensed, it is transcriptionally no longer available, right? Transcription factors can no longer see the different DNA motifs that they recognize, so you don't attract RNA Pol II there. Right? So let's see what happens during the development of cancer, for example. Right? Here you have a gene. This is this RAS SF1A. It is a gene. This is not RAS. Okay, this is a different tumor suppressor protein. And it is normally expressed from this gene here. So here's exon 1 of this RAS F1A tumor suppressor. Right? So what they've done here is they've taken a whole bunch of cells from normal tissue from a patient, okay, and they've done what they've called a DNA methylation sequencing, where they've identified that in this region, there are multiple CPG sites in this promoter. So you can see there are 22 CPG sites here, and this is how they're kind of spread out in here. So each one of these little dots represents the status in one of the cells. So each of these lines represents a molecule of DNA taken from a particular cell. In normal control tissue that's healthy, you can see that none of the CPG sites are methylated. They're all white, right? They're not blue. So therefore, right, this RAS SF1 gene can be easily expressed, right? So it makes this tumor suppressor protein that's doing its job and trying to prevent the cell from dividing out of control. Let's look at some samples from a tumor. There you go, right? So you can see from numerous tumor cells, they've looked at the CPG methylation status, and you can see the promoter region of this tumor suppressor gene has become heavily, heavily methylated, right? So therefore, in these cells, they no longer express this important tumor suppressor protein. Okay? What about the cells that are in normal tissue, but right up against the tumor tissue? That's kind of interesting, huh? What do you think it looks like? It looks like this. You begin to see the evidence of certain transforming events happening in these cells, because these cells are a mix, right, of tumor cells as well as normal cells. So methylation status of tumor suppressor genes, not surprisingly, can be a very important way to distinguish which tumor suppressor genes are the important ones when you're talking about one type of cancer versus another type of cancer. This is a wonderful figure here, because what they've done here is they've taken an array of different important tumor suppressor genes on the right over here. Right? Some of these are ones that you may recognize. For example, Here is APC, which we had talked about previously. We'll take another closer look at APC. Other proteins such as BRCA1, I know some of you are aware, BRCA1, right? That plays an important role in certain gynecologic cancers like breast cancer or ovarian cancer. And what they've done is they've looked at the methylation status of these tumor suppressor genes taken from tumor samples of these different varied cancer types. And what this allows you to see is that this set of tumor suppressor genes are not equally important for one cell type and one cancer type versus the other. For example, BRCA1 in, let's say, breast cancer tumors, they see elevated evidence, right, of BRCA1 methylation in breast, ovarian, uterine cancers, mainly sort of gynecologic cancer, gynecologic cancers, right? That means in those tumors, you tend to see that the BRCA1 gene has become methylated, right? And the BRCA1 tumor suppressor protein is not being made properly. Whereas for others, like this CDH1, for example, right, it seems like it is almost like the most important tumor suppressor involved in things like, what do we have here? Esophageal cancer, right? Or over here, heavily involved in breast cancer as well. Right? Other tumor suppressors are more sort of general. For example, here's one called P16. Right? And you can see P16, all of these different cancers, all of these different tumor cells that were looked at, there's evidence that you see P16 gene methylation, right? So the shutdown of P16. So what I want you to sort of have as a take-home message from this, I don't expect you to memorize this, whatever. I do expect that some of you are interested in this and you'll look at this a little more closely, right, to see some evidence of which tumor suppressor gene seems to be most important for particular cancers that you're interested in. But the take-home message is really that certain tumor suppressors, like the P16 gene, right, its loss due to methylation is common to many cancer sites. Whereas other tumor suppressor genes, right, the expression of their genes is lost in more specialized cases. BRCA1 loss is mainly seen in breast and ovarian cancer. APC loss, the one way over here, is mainly seen in different types of GI tract cancers. Right? So this methylation, how does this happen? Right? How do you actually end up with cells that start improperly methylating the promoters of tumor suppressor genes? Right? Here is a slide providing you one part or one piece of evidence. There are enzymes that are in charge of adding methyl groups to CpGs. So these are the DNA methyltransferase family of proteins. We have DNMT1, DNMT3. In this case, we're looking at DNMT3B. Right? So this protein, if it's allowed to interact with the CpG sites of certain promoters, would start transferring methyl groups to those. Here we have 3 panels looking at different stages of cancer. In this case, I think they're looking at colon cancer. So this is the epithelial lining or the glandular lining of your colon, right? And the epithelial cells that make up the lining, many of them are glands that make things like mucus, right? And so what they've stained here are in blue is the production of the mucin protein by these cells, right? Because that's what their job is, to make this mucin protein. And in brown, they've stained for levels of this DNMT3B. So normally in healthy normal tissue, right, these glands are doing their normal differentiated cell function, so they're making tons of this blue stained mucus, right? And they have very modest levels of the DNMT3B protein in them. As they transform and start becoming a tumor, here you see a less aggressive tumor, right? So maybe this is not necessarily cancer yet, right? But it's transformation. What do you see? You see the cells clear evidence that they're beginning to lose interest in doing their normal differentiated cell function, right? Because they're producing less of the blue stained mucin proteins. And of course, the striking thing is levels of DNMT3 protein are going up in these cells, meaning more and more methylation of different promoter sites in genes. A very aggressive tumor, maybe now this is full-fledged cancer, you see these cells have completely given up their normal differentiated cell function. They no longer make mucus, right? And they have a ton of DNMT3B in them, meaning the methylation of their genes is really out of control, right? So you might say, well, but then wait a minute, because couldn't this result in the methylation of proto-oncogene promoters? Yeah, it could, right? In the same way that you might have out of control methylation of proto-oncogene promoters, you would have methylation of tumor suppressor gene promoters. It's just what combination is taking place inside of these particular cells, right, is leading them to become tumorous. So let's take a closer look at how tumor suppressor proteins that are made from tumor suppressor genes, how do they work? There are two sort of main functional ways of thinking about the importance of a particular tumor suppressor gene. Some of them are more like supervisory, some of them are more like hands-on. So you say, wait, that's kind of like the same way we talked about proto-oncogenes, right? Some of them are more supervisory and some of them are more hands-on. Exactly, right? But in this case, right, we're saying these supervisors and these hands-on proteins, right, they're working to try to stop the functions of oncogenic pathways. Whereas the oncogenic pathways, their supervisors and their hands-on workers are driving oncogenic potential. So tumor suppressors, we can call them gatekeepers and caretakers. Gatekeepers are ones that make broad decisions about housekeeping, right? So for example, if you think about a hotel and the housekeeping crew at the hotel, the gatekeeper people might be the manager, right, of the housekeeping team. So he or she makes broad decisions that need to be done. Like this week, we're changing all the sheets to this new pattern, or this week we need to take down all the curtains in this wing of the building to get them laundered. Right? So they're making broad decisions based upon different signals. And ultimately, these gatekeepers prevent the cell from proliferating if the signals are not right, in order to allow the cell to go through the cell cycle, right? So if those signals are not right, the gatekeepers make broad decisions, and those broad decisions are carried out by the caretakers, right? So it's one thing for this housekeeping manager, supervisor to make the decision about what to do, but it's ultimately the hands-on caretakers who do the things like changing all the bed sheets, taking the curtains down, etc. So what does that mean? Remember in the previous slide where we looked at that colorful three-dimensional thing of all these columns of data, looking at some tumor suppressors being very specific to particular types of tumors, whereas other tumor suppressors, loss of those seem to influence a whole bunch or almost all the different types of tumors. You can now begin to appreciate, right? Things like BRCA1 or APC, right? Loss of those can have more specialized consequences, whereas loss of something like P16 would have broad implications, just as P16 would be considered one of these gatekeepers, whereas BRCA1 and APC, especially APC, right, is more of like a very specific tumor suppressor caretaker. Right? So we ended up evolving many diverse pathways of tumor suppressor genes. There are far more tumor suppressor gene pathways in our cell compared to proto-oncogene pathways. You know, proto-oncogenes, we've talked about ones like RAS, SARC, we've talked about things like MYC, right, and AKT, things like that, right? Those are like real standouts. Okay, and those kind of represent the main proto-oncogenic pathways in our cells. And yet, as we will see, tumor suppressor gene pathways are much more diverse. So why is that? Why have we evolved much more diversity in the TSG pathways compared to proto-oncogene pathways. What would be the evolutionary rationale for that? Well, it has to do with this. Right? Good brakes are more important than good acceleration. Right? What do I mean by that? You have your car, right? Your car has its own set of accelerating mechanisms, which, if you think about it, is really just your gas pedal, right? But you have numerous ways of making your car stop, right? You have your normal brakes, you have your hand brakes. I mean, you could extinguish the engine, right? Why is it more important to have good brakes in your car than it is important to have good acceleration? It's because if your acceleration is kind of compromised, let's say you're having trouble with your fuel injection or you lost a few spark plugs or something like that, right? And your car is much slower to accelerate, you can still use it to get around, right? But what happens if you lost your brakes? If you lost your brakes, it could be catastrophic. Right? So therefore, we evolved many more braking pathways to make sure that cells don't lose control over cell proliferation. So what we'll do is, for the rest of today's lecture, we'll look at just kind of a little sampling of some different examples of how diverse these different tumor suppressor gene pathways can be. Right? And this is just a small sample of many numerous possible examples. We already looked at P10 and we looked at RB. We'll take a closer look at RB when we really look at the cell cycle. Okay, and we'll look at things like P16 as well. But for today, let's look at these three examples. These are tumor suppressor genes that make tumor suppressor gene proteins, right? So NF1, APC, and VHL. So let's start off with NF1. So NF1 is a TSG that helps regulate the RAS signaling pathway. So NF1 is a protein that has in its structure the ability to interact with different, so it functions as a gap, right? Remember what we learned what a gap is? A gap is a activating protein, a GTPase activating protein, right? It is in charge of encouraging RAS to turn off. Remember, RAS, when it's bound to GTP, it is active, right? But RAS has the ability to cleave the GTP to turn the GTP back to GDP, in which case RAS is turned off. And we already learned the longer RAS stays in its GTP bound state, right, the longer it can signal. And therefore, the stronger the oncogenic signals are that RAS is driving. So gap proteins encourage the RAS to just go ahead. You know, hey, stop this whole oncogenic signaling thing. Go ahead and do your job, which is to actually cleave the GTP, right? So gaps stimulate the GTPase activity of RAS. So proteins like NF1 function as a gap because they have in their structure this particular arginine finger. So it is a structure here that actually, when it binds to RAS, this is the region of the protein that kind of gets RAS kind of to change its shape a little bit to encourage RAS to go ahead and cleave the GTP, right? So what does that mean? That means the more NF1 activity you have, the less RAS activity you'll have. And that's how NF1 is functioning as a tumor suppressor protein. Mutations in this NF1 can have a profound effect on this. So the critical residue here is this arginine at position 1276 in the NF1 protein structure, because this is the arginine that inserts into the RAS GTP cleft and encourages RAS to break the GTP, turning off. Mutations at that arginine location, right, have the ability to reduce the NF1's gap function directed at RAS by over 1,000-fold. Right? So you really eliminate the ability of NF1 to be able to interact with RAS to properly manage RAS. So not surprisingly then, right, when you look at lots of tumor samples and you look for what mutations in NF1 might be relevant, time and time again, you see that many of those tumor samples will have a mutation at arginine 1276. Right? And why is this called NF1? Because the particular type of cancer that this protein was first discovered in as being important was a neurofibro-related cancer. Right? So individuals who are NF1 heterozygous plus minus exhibit a phenomenon that we call haploinsufficiency, meaning even though they are still plus minus heterozygous, the fact that they are only down to one good copy and that they have one defective copy, right, is not enough because they are predisposed to getting the disease. So this is the disease, this neurofibromatosis type 1. So it's a common familial cancer, right, because it has to do with things like mutations in the NF1 gene that gets passed on through the generations. It affects about 1 in 3,500 people worldwide, and it's a syndrome, meaning it's not one particular type of cancer, but many related symptoms. So the development of tumors, they usually involve the sheath cells that surround neurons. And so if this has gone unchecked, then these might develop into neurofibrosarcomas, right, tumors. So oftentimes you see this as these what they call cafe au lait spots on the skin, right? So these are regions where cells have are beginning to kind of transform, or they're not functioning properly, or these nodules in the eye, right, representing the formation of tumors on nerves in the optic nerve. Okay. Our second example is APC. So APC, we've learned already, is an important regulator of beta-catenin. And so APC, I think I told you already, is called APC because it was first discovered as being highly important in colon cancer. So adenomatous polyposis coli is what APC stands for. So as a quick review, remember we said that beta-catenin is an important transcription factor for activating certain genes that drive the cell cycle forward, right? So this would be considered a proto-oncogenic pathway. Normally, our cells control this by destroying the beta-catenin. So on the left, normally beta-catenin is held up by this big complex of proteins that include this GSK3 beta that phosphorylate the beta-catenin. The phosphorylated beta-catenin is degraded before it ever has a chance to get into the nucleus and activate target genes, right? Normally, the signaling by Wnt, this red thing from the outside of the cell, signaling the frizzled receptor, results in the frizzled receptor disassembling or preventing the formation of this complex that destroys beta-catenin, and therefore beta-catenin is allowed to escape, right? And that's what signals. So Wnt signaling then, arriving at the cell, is a signal that drives beta-catenin signaling and drives the cell to proliferate. Right? And where's this Wnt signaling coming from? Well, if we're talking about this is an epithelial cell, right, the Wnt signaling would be arriving from those connective cells in the stroma that send signals to the epithelial cells to tell it what to do. You know, yeah, you're in a good spot. Go ahead and do your normal epithelial cell function. Don't worry about dividing. But when it's time to divide, here's some Wnt signals, right? And the Wnt signal arrives on the epithelial cell, and that results in beta-catenin activity going up, and now the cell begins to proliferate. Right. So one of the important components of this beta-catenin degradative complex is this APC. Right? So if APC is compromised, right, then APC will not be able to properly help out forming this complex, and as a result, depending upon the nature of the compromise in the APC, right, you might have out of control beta-catenin signaling. It'd be more like this side if this APC, let's say, is the wrong shape or the APC is no longer there. Okay. So this is very important for understanding things like colon cancer development. So remember the lining of our colon, if you were to really zoom in, right, our colon, of course, is very sort of, has many folds to it because the folds of the epithelial layer maximize or dramatically increase the amount of surface area for things like water, to kind of go back and forth to be absorbed from your food, for example, right? But even if you zoomed in on those folds, you would see that the cells that make up these folds have many of these little valleys, which are our colonic crypts. The epithelial cells out at the top of this would be the ones that are really exposed to your food that's coming by, right? So these epithelial cells are shed all the time, right? Like a huge percentage of the solid waste that your body gets rid of is actually dead epithelial cells that are being shed off by the fact that physically, as the food's moving by, it's just scraping these cells off, right? So that would be at the top here. Right? Where do these epithelial cells come from? Well, they come originally from the bottom of the crypts. Right? So the bottom of the crypts, you have these stem cells in gray. And these stem cells are in charge of proliferating to repopulate all of the epithelial cells that are being sort of sloughed off in your intestines. So these stem cells will proliferate. And remember, stem cells, they're not concerned necessarily about being a gland or having cilia or something like that. Stem cells just divide, right? They're just trying to replenish the number of cells that then go on to differentiate through additional divisions and additional sets of gene expression waves to become the epithelial cells. So as they divide from the bottom, they're dividing because they are receiving Wnt signals, right, from these stromal cells that are on the other side of the basement membrane. That's not shown here, but these red arrows are like the Wnt signals that are arriving and stimulating the beta-catenin signaling to take place. So as a result, these cells are just dividing, and they kind of get pushed up the walls of the crypt as the stem cells at the bottom keep dividing. Right? So as long as they are still receiving these red arrows, the Wnt signals, right, they are happy to just divide. At a certain point, they are physically far away from these original Wnt signals that they no longer receive those Wnt signals. And so now the beta-catenin, right, starts to get more carefully controlled. So on the left side here is what you would see. You would have sort of this imaginary line, this dotted line, right, below which the cells are still preoccupied with dividing because the beta-catenin signaling is on. But once they get far enough away and they no longer receive the Wnt signals from the bottom of the crypt, from these stromal cells, right, they begin to shut down their beta-catenin signaling, and as a result, they shift to becoming more differentiated until eventually they become a full-fledged, fully differentiated colon cell that then can do its job before then just falling off, right? I think a typical epithelial cell in your colon lasts maybe like 3 days or 5 days once it gets up here before it's shed. Let's see what happens on the right, though. Okay, on the right, you have these cells that are responding to Wnt signaling, so they're dividing. At this point, they should be far enough away that they no longer receive the Wnt signaling, so they should be starting to, divide and not stop dividing and be more about differentiating. So some of them are, but look what happened. One day, somewhere along the way, right, a cell took a hit or took a couple hits in its APC gene, right? So now the APC gene was eliminated. These cells without the APC don't turn off their beta-catenin signaling. And as a result, they just keep dividing with little interest in becoming fully differentiated. So this then becomes the start of a tumor. Okay, yeah, so this is, I know this is like right before lunch. You probably don't want to see this. But this is a good example of what adenomatous polyposis coli looks like. Right, so this is a normal colon cell wall here. So it's really folded and ruffled again to maximize the surface area. Right, so people who have a disease known as APC, which is also called familial adenomatous polyposis or FAP, right, they develop tons of colonic polyps. Right, so these are individual little tumors that are not necessarily cancer. Right, because remember, these are just tumors that represent hyperplastic tissue. Right? About 50% of people who have APC develop these types of lawns of polyps by the age of 70. Right? And certainly there's a much greater chance of these people having one or more of these polyps progress to cancer, because most colon cancers are sporadic. About 95% or more of colon cancers that present in the clinic, right? There is no evidence of any kind of inherited defect in tumor suppressor genes like APC, right? But people who are born with bad alleles of APC, they will have inherited a much elevated increase of risk of developing malignancies. So APC is widely studied as a popular model for understanding how tumor suppressors function inside cells. And so there is a mouse model that has been generated that's known as an APC min mouse. And so these mice are engineered. They have a particular mutation or an allele in their APC gene that forms a truncated APC protein, right? So this APC protein is no longer the right shape, so it cannot participate in forming that beta-catenin regulatory complex. And as a result, the beta-catenin doesn't get degraded, and the beta-catenin is allowed to keep working in these cells. So this truncated form of the APC gene, they call it MIN, or multiple intestinal neoplasias, right? Because that's what they see resulting in these mice. And these mice who are born with this mutation, right, they spontaneously develop colon tumors. And that's why they are a very popular model for studying colon cancer. Here you can see, here is some tissue from the colon of one of these APC min mice. And what they're staining in red here is beta-catenin, right? So wow, I mean, this really hits you over the head. There's no doubt that the levels of beta-catenin in these cells is just out of control, right? Because they don't have APC as an important tumor suppressor gene to help keep the beta-catenin in check, right? So beta-catenin is not being degraded. It always just builds up. Here's a question. These APC min mice are technically min/plus heterozygous. Right? Why are they not bred to be min-min? Right? Why are they bred to be heterozygous and not homozygous for the defective allele? Hopefully, if some of you are thinking about this, you realize because APC is such an important tumor suppressor gene, that even from the early development of the embryo, you need to start worrying about controlling things like beta-catenin. And so if the mouse was engineered to be completely APC knockout, right, that it didn't have APC at all, such a mutation would be embryonically lethal, right? And the developing embryo would never make it to full development and being born. Right? So these mice then are engineered to be heterozygous so that they can be born, but ultimately they will spontaneously develop colon tumors. Our third example that we'll end with today is this VHL. All right, so VHL is a protein that helps regulate our cells' responses to oxygen levels, in particular, low oxygen levels or hypoxia. So VHL was named after this disease, von Hippel-Lindau disease. Right? So it's a syndrome that people who are born with VHL, they're predisposed to higher risk of developing tumors in breast, kidneys, adrenal gland, even certain types of blood vessel defects come from it. It affects about 1 in 35,000 people in general population. Almost all VHL patients, the problem can be traced back to defects in this VHL gene, right? That was identified as the gene responsible, and that's why it's called VHL. What does VHL do? The protein that comes from this gene, so the protein VHL, degrades another protein called hypoxia-inducible factor 1 and 2, right? So HIF1 and HIF2. These proteins are very important in regulating a cell's response to low oxygen levels. So here we have an illustration of how this works. So you have these proteins such as HIF1 or HIF2, that are used as sensors in a way to detect whether a cell is getting enough oxygen. So this HIF1, the phosphorylated HIF1, if there is normal levels of oxygen available for the cell, so under normoxic conditions, it's kind of a funny word, right? There is a protein called proline hydroxylase. Right? These are not phosphate groups, sorry. These are proline residues, right? The proline hydroxylase takes oxygen, right, and puts these hydroxyl groups on these prolines here. So that HIF1 protein that has these hydroxyl groups added on because there was plenty of oxygen in the cell so that this proline hydroxylase could go ahead and do that. It is the right shape for this tumor suppressor protein known as VHL to bind to the HIF1. And as a result, the VHL facilitates the addition of ubiquitin groups. So ubiquitin is a small protein that can be added on to other proteins to serve as a label or a flag. Right? Quite oftentimes, ubiquitination or ubiquitination of a protein leads to that protein being degraded by the proteasome. Right? So in other words, if your cell is getting plenty of oxygen, right, the HIF1 is destroyed by the proteasome. However, under hypoxic conditions, in other words, low oxygen conditions, the cell might start getting stressed out. It's not getting enough oxygen. As a result, HIF1 is allowed to function because under hypoxic conditions where there's not much oxygen around, it's not going to have these hydroxyl groups added to it. So as a result, it does not get targeted for degradation. The HIF1 is allowed to escape being bound up by the VHL tumor suppressor protein. The HIF1 is allowed to go and function as a transcription factor. So it goes into the nucleus and turns on certain genes that the cell wants to turn on and make proteins in response to low oxygen conditions. And some of these are genes like VEGF. Right? VEGF is a protein, or the protein product of this is very important for encouraging additional blood supply to feed the tissue. Right? So the development of more capillaries growing into the tissue to supply higher levels of oxygen that's badly needed. Right? So people with defects then in the VHL means in those people, the VHL never has a chance to properly lead to HIF1 degradation. So HIF1 is allowed to function as though you're under hypoxic conditions, right? And as a result, tissues where this mutation of the VHL tumor suppressor gene is taking place will have things like more blood vessels supplying the tissue with oxygen. All right, so this is the last slide. In familial diseases, why do we see that mutant tumor suppressor gene alleles like retinoblastoma or NF1 or VHL or APC, these are the things that get transmitted through the germline, right? Familial cancers linked to mutations that are passed on almost always involve mutations in tumor suppressor genes. You very, very rarely see that the mutation is traced back to a mutation in an oncogene. In other words, mutations in SARC, MYC, and RAS, right? You never see these as mutations that are passed on through a family. Why is that? Why is it that it's the mutant TSG alleles that are transmitted through the germline, right? But oncogenic mutations in proto-oncogenes, right, are not typically passed on? It is because TSGs work in diverse overlapping pathways, right? So even if you have a defect in one TSG, right, you have many other TSGs that can still kind of work and allow the embryo to still develop, right, and become a person who's carrying that hit in retinoblastoma or APC or VHL, etc. However, with oncogenes, if you were to have a mutation that results in an oncogene acting out of control, like a RAS mutation, right? If that was passed on to the embryo, that embryo would never properly develop and never reach the point where it can be born, right? So that's why another important evidence that we evolved multiple tumor suppressor gene pathways, right? Because that is really the emphasis that Mother Nature wanted our cells to worry about, right? Is keeping the brakes ready and functional, right? So that when the acceleration starts getting out of control, you have different ways of stopping cell proliferation. Okay. All right.